Good morning. Today, I'm going to share my stance on the ethics of clinical therapy. But first I'm going to ask, would you want your dying brother to participate in a clinical trial and receive a placebo treatment instead of medicine that actually had a chance to cure him? The answer is obviously not. But in the name of progressing science and figuring out whether a treatment actually makes a difference, placebos are part of clinical research. Incurable diseases mandate a different set of ethics than other diseases do, and that's what I'm going to talk about today. So I want to start out by introducing you to Ethan.
Mystery. Ethan, about a year and a half too late, got to the doctor's office and discovered he had ALD. He's a 9-year-old boy in this picture with braces, thinking about the future, thinking about whatever a 9-year-old should think about. But in that instant, when they went to the doctor, and in this case, a year too late, they found out he had ALD. In this picture, which was less than 6 months, from left to right, he's lying in his bed, and this was the last picture ever taken of Ethan. He died 3 months after this picture, at the ripe age of 10. So if you're not uncomfortable, you're not human.
Gene therapy could have been the answer for Ethan, and this is why we need to move forward exploring gene therapy in clinical trials to save people like him. One of the first clinical trials was Jesse Gelsinger in '99. He had a genetic disease, so doctors at UPenn injected him with a virus carrying a perfectly engineered gene in hopes that the virus would detect the defective genes and correct them. When Jesse died 4 days later from a massive immune response, he became the first person publicly identified having died in a clinical trial for gene therapy. Now, the Gelsinger case brought forth 3 inexcusable flaws in the realm of clinical trials. The first issue was something called publication bias. Researchers withheld the fact that monkeys had died after being given a similar treatment. The second problem was a huge error in informed consent. Doctors are required to disclose possible risks and benefits verbally and in writing, but in this case, 2 patients had had serious side effects, and that appeared nowhere in the informed consent document. The last big problem were that financial incentives clouded the head researcher's judgment. He was looking to patent this gene therapy delivery system and make millions. So first, I want everyone to understand the science. When I talk about genes, I want you to picture an orchestra. The cells in a person's body act in concert. The nucleus, or the brain of a cell, contains a person's genome, which is about 80,000 cells. Every cell has every gene. And genes tell the body how to behave, what proteins to produce, when to grow. They're the written instructions or the music that cells play in different amounts at just the right times. And the result is a complex symphony. Some airs, like a violinist playing a flat note, aren't a big deal. But other airs are more profound, like a symphony that never started. And that's where we run into trouble with genetic diseases. Now, lucky for you, genes are found in long strands of DNA, which is deoxyribonucleic acid, like a thread around a spool around several proteins that looks something like a gummy bear. So now you can open your bags and chew on some gummy bears. I know it's a little early for that. And what you're chewing on is a chromosome. So we've already talked about the most important people in the issue, the patients. The people like Jesse and Ethan. But we also have all these people fighting for control. Scientific discoveries may push for one ethical decision in a clinical trial, while responsible regulation by the FDA may depend on another one. A high percentage, in fact, of all drugs tested on humans never make it through the FDA. And companies lose tons of money each day, and new drugs approval is held up. The financial ambitions of pharmaceutical companies, not to mention personal greed, should never be able to overshadow the safety precautions in clinical trials. The primary reason for developing gene therapy is to help people with genetic diseases. While society's approach to disease should be to prevent it wherever possible, certain rules must apply to prevent the dark world state that Aldous Huxley criticizes in his science fiction novel, Brave New World. In the book, 10 world controllers apply the principle of mass production and perfecting genes to produce predestined, socialized human beings. Sure, the people don't have any diseases, but that comes at the expense of the brainwashing and hypnotization that control everything from their chemical makeups to their sex lives. And I would argue that to a certain extent, genetic errors are part of genetic diversity. Now, gene therapy's role can be to fix the very large genetic errors. If you remember, I'm talking about the errors that result in a symphony never starting. That saves people like Ethan and like Jesse. Gene therapy should strive to make this happen as long as all the members involved agree to prudently abide by what I see as the ethically sound codes of conduct. Society needs to engage in a discussion and set ethical boundaries for what make transparent, fair, voluntary consent agreements, so no single profession has the final decision. Now, how are those gummy worms? We're going to go back to the science for a minute. I'm going to show you how to fix cells with gene therapy. So I'm talking about blood stem cells, the ones that make your immune system, your central nervous system, your blood. In a science lab, a specifically engineered virus is added to a patient's blood and then returned to the body to fix the problem.
To do is to infect
This is the
the
virus.
cells. Infect it as many times as we can, as many cells as we can in that bag.
This is all taking
Goes
place in
into
the bag.
First the cytoplasm, then it releases the payload. It then knows to, and we're certainly taking some scientific liberties here to make it a little more simple, but it explodes there, right there. That's your mothership, the RNA.
That's your
For
RNA.
those of you who remember your biology class, we got to do some gymnastics to make sure that turns into DNA. Because we have here, up here it says revolutionary ideas.
This is where all your genes are.
Watch carefully here, because as we finish this gymnastics, you're going to visualize the revolutionary idea. This gets moved and transported into the blood stem cells compartment, the nucleus, where the
Going into the
DNA exists. Right here, a little magic, boom, it integrates itself into the DNA.
This is the fixing part.
Now, what you've just created is a gene-modified blood stem cell. That stem cell knows what to do. The cells know what to do. It starts producing your therapeutic gene. It starts producing a lot of them. You turn your red cell into blue cell in this picture here. Bottom line, those are modified. Those now know how to do what the problem was before.
So that's gene therapy. Now, take the paper out of your bag to find out whether your 19-year-old woman who will soon suffer from genetic blindness, I'm going to refer to that as LCA, the father of a child with bubble boy syndrome, I'm going to refer to that as SCID, or a 65-year-old man with Parkinson's, who happens to be my grandpa. Now, I'm going to pretend to be each of them to show you my stance. First, I'm the girl. My doctor just told me that a total of 9 LCA patients underwent gene therapy treatments and saw significant improvements in vision. LCA isn't deadly, so gene therapy wasn't initiated on the patients until they were 18 years or older, old enough to give consent on their own. I believe that because blindness prevention is a quality of life rather than a life or death issue, I should not have had to wait until I'm 18 and now my disease has progressed to almost blindness before I can do experimental gene therapy. My parents should have been able to give consent for me to participate in clinical gene therapy because it has been proven that I would have had a greater chance if I still had more light receptors left in my eyes. I would have wanted the best shot possible to fix my LCA, especially because the worst outcome of treatment failure would have been blindness, which is the natural outcome of my disease. The ethical principle of not treating patients below the age of consent delayed my gene therapy. Next, I'm going to be the parent of a child with SCID. As a parent, I believe I have the right to enter my child in a clinical trial, just like I have the right to provide consent for circumcision, which is an irreversible change to bodily structure, preventative vaccinations, and surgeries that sometimes make children permanently sterile. I'm entering him in a clinical trial because if I don't, I'll never be able to take him outdoors because his immune system can't handle germs. I would rather live with him inside for all his life than live with him not at all. But I want to know that risky trials like the one Gelsinger was put through are reserved for people who otherwise don't have a chance. My son has a chance, and I will not tolerate the cherry picking of data by researchers whose first priorities are fame or money. I listened to the TED Talk, What Doctors Don't Know About the Drugs They Prescribe, and I've learned that research trials need to be properly registered. They need to share their exact plan with clinicaltrial.gov, and all trial results need to be published within a year, whether they are positive, whether they are negative, and not only trials after 2008, all trials. Last, I'm going to be the grandpa. My doctor informed me of a gene therapy trial that may help my body relax and help my Parkinson's, but he does not have large numbers of treated patients to point to. So I'm going to hold off on treatment until more data is obtained from clinical trials, using more desperate patients with advanced stages of the disease that have already been treated with conventional standards but progressed nevertheless. While successful treatment of my Parkinson's is dependent on the number of surviving nerve cells, and my nerve cells are going fast, I would rather live these next 5 years to see my grandchildren graduate high school than die participating in early stages of a dangerous trial. But on a side note, testing gene therapy on consenting prisoners is a very viable option. Some ask whether it's appropriate for prisoners to give informed consent when they don't even have the autonomy to decide when to eat, but with adequate